All right, hi everybody. I'm Caitlin. I obviously collaborate with Claire, so I'm going to be talking about the SERS detection of the chemistry. So she has already gone through a lot of the prostate cancer background and the chemistry needed to find the detection molecules, so I'll be talking about the detection itself, obviously using photonic crystals to enhance SERS signals. So first, we'll go into a little bit of why we need this. Claire already talked about it. We'll go into a little more, but then the majority of my presentation will be a background on photonic crystals, a little more information on SERS, and then we'll get into some of the data we've collected. So first of all, the motivation for this is, as Claire mentioned, the current detection for prostate cancer using PSA isn't all that reliable. So what we want to do is we want to find something that we can get lower limits of detection and more reliable detection of something like zinc or sarcosine that we can use instead of PSA for prostate cancer assays. So what we plan to do is use the electromagnetic enhancement capability of photonic crystals. One example is if you use these use these with fluorescent assays, there are lower limits of detection than more traditional methods like ELISA, and as we'll also show, there's a lower limit of detection for the traditional method of using SERS on glass surfaces. In this one example for prostate cancer is part of a larger effort to create low-cost portable biosensors. So the idea is you get a small sample of liquid and you have a portable detection system that you can take to something like a third world country where they don't have access to laboratory facilities and they would like cheap screening methods to reduce death rates from cancers and other viruses. So this is a photonic crystal. It's fabricated in a clean room on a silicon wafer. So on top of the silicon are two dielectrics, the silicon dioxide and the titanium dioxide, and they have titanium dioxide is a high refractive index and silicon dioxide is a low refractive index. So what happens when you stack those two on top of each other, if you hit it with the right wavelength at the right angle of light, it creates a resonance condition, and the light will be trapped inside the silicon dioxide cavity, which creates more energy in the system. This is shown on the graph on the right. You can see the red spots, we call them hot spots. They're at the corners and the interfaces. This is where the light is trapped and is reflected back out. I guess we have air on top of the titanium dioxide. This effect also depends on what medium you're sitting in. So the wavelength and incident angle condition will be different for if you're in water or you have proteins attached to the surface than it will be for air. There are also two different methods of enhancement for photonic crystals. One of them, which I just talked about, was enhanced excitation. So that's the incident angle and wavelength condition. That's what's shown on the right. The other one is enhanced extraction. So the light that comes out of this, the light that's reflected out of the crystal is all perpendicular to the crystal. Normal scattering on glass would radiate in all directions, but for photonic crystals, the majority of it is perpendicular. So what happens is you get more efficient collection of the light by your detection system. So this is what I was talking about earlier about the angle dependence. So if you have a photonic crystal, there are two directions. One of them is horizontal, the other one is vertical. On the graph on the left, the theta direction, that's parallel to the grading lines, which, these would be the grading lines. So parallel to that direction, the angle doesn't matter too much, but perpendicular to the direction, we get very strong dependence. You can see this is wavelength, this is angle. So for whatever wavelength you're using, you're going to want to change the incident angle to get the maximum enhancement. And this is just an example. On the bottom here, this is a photonic crystal at normal incidence. Notice the wavelength is different than when we change the angle to 3.5 degrees. And what we can also do is for a given wavelength, this would be illuminating the photonic crystal with a laser, we can do for a given wavelength is find out the incident angle. So you can do it both ways. And this is the custom hardware we have set up on our lab that we use to do photonic crystal scans. There are some important parts. So there's a laser over here. It goes through a cylindrical lens, which focuses the laser beam, which is a collimated beam, which is just a solid block of light that doesn't expand or focus as long as it goes. What it does is it focuses the collimated beam to a line, and that way we have a line of light that's perpendicular to our grating. So that gives us most control over the angle and the most enhancement that we can get from this system. So the line is focused onto the photonic crystal, it's reflected back, and the reflected light passes through the mirror, through the emission filter, which filters out the laser light. So we don't want extra noise in the system. So all the reflected light that we care about is at different wavelengths than the laser. So if we filter out the laser light, that helps us a lot in our detection. So passes through the emission filter, goes through the lens, and inside this box for the CCD camera, there's a grating. So if you, there's a diffraction grating. So if you illuminate light on the grating, what you get on the other side of the grating is a spectrum. So then we capture the spectrum on a CCD camera and analyze that to get the Raman spectrum. So that was a little bit on photonic crystals. So now we mentioned how you get the Raman spectrum. So we'll talk about what SERS is and why you would want to do it. So first of all, Claire went into the fabrication of gold nanoparticles. Why do we want to use gold nanoparticles is a good question. For biological applications, there are two main metals that somebody would use. It's gold and silver, because for biological applications, we're interested in light in the visible regime. So gold and silver both have plasmon resonances in the visible regime, but the reason we use gold is because it's not as reactive as silver. So silver could degrade under oxygen if you just let it sit around. It could interact with your samples in a bad way. So gold is not as good of resonator as silver, but it's our best option. So as Claire talked about a little bit, it's called the local surface plasmon resonance. So this wavelength is dependent on the size of your gold nanoparticle. So these four are differently sized. Oh, sorry, this is clusters. So these are single clusters, double clusters, quadruple clusters, and then just randomly scattered particles. So the resonance depends on the size of your clusters, the size of your particles, and how far spaced apart they are. So as you can see, these 1, 2, 3, 4, each of these has a different resonance wavelength depending on the size of your clusters. So for best control, you kind of want monodispersed single clusters pretty equally spaced on your surface. It turns out to be really hard to achieve, but you would want that to get like the tightest peak in your resonance angle. So this is the peak wavelength of these different gold nanoparticles. And then this is the Raman shift. Sorry, this is the SERS spectrum for each of these different ones. And you'll notice that the largest SERS spectrum has to do with the gold nanoparticles that couple most closely. And this one is a similar effect, except it has to do with, which one is this? This is looks like the spacing of the particles. So these are really far spaced apart, and these are all tight together. So as you can see, as the spacing changes, so does the resonance wavelength. Yeah. And so does, I guess this is the intensity with the particle diameter. So spacing, cluster sizes, and particle sizes all affect the resonance wavelength that you're looking for. So this is what happens if you put nanoparticles onto a surface. So these would be some of your gold resonators. And again, this is the hotspot that we talked about before. The photonic crystals have hotspots in the corner. The gold nanoparticles have hotspots at interfaces with the surface. So interfaces with the surface and also interfaces between particles. So the closer particles together, the more hotspots you're going to get until you get them too close together, in which case they either form clusters or they quench each other, both of which are bad. But as you can see, you get larger enhancement for tighter spacing. So what happens if we put the photonic crystal together with SERS detection? You can see this graph, which I talked about earlier. You get more enhancement with photonic crystal SERS than you do with the glass, which is typical for SERS, which is just in black. So on resonance, you're going to get both enhanced excitation and enhanced extraction. Off resonance means that you detune it from the on resonance angle, so you still get a little bit of enhancement due to enhanced extraction. So instead of glass, where the light radiates in all directions, the light from the photonic crystal will go towards your detector. So that is good, even if you're not able to couple it to the resonance angle. And the other note to say is here is an example. Claire talked about symmetric gold nanostars. So it turns out the particle enhancement for symmetric particles is also larger than for asymmetric particles. So there's a lot to consider when we're trying to get gold nanorods on a photonic crystal to get enhancement. This dark field microscopy is a step that I do after I get the gold nanoparticles to find their resonance. So typically with the photonic crystal, what we can do is on the left, we can just take the photonic crystal, shine white light on it, and measure at what wavelength use a spectrometer to measure at what wavelength the intensity is the highest. That's all fine, but it turns out that gold nanoparticles are too small to affect the bright field reflection, so we have to use a dark field microscope, which measures the scattering from gold nanoparticles. So what we do is the scattering is a measure of the local surface plasmon resonance. So the wavelength at which the highest scattering occurs is the same as the wavelength we're interested in. So we use a dark field microscope to get the resonance of the nanoparticles, coupled with the bright field microscope, or the bright field reflection, to get the resonance of the PCs. So this is just an example of dark field microscopes, or dark field microscopy. You can see these tiny little dots, and then each of which correspond to a different resonance wavelength. So these dots are going to be different nanoparticles of different sizes. So you can use that to measure the resonance wavelength. So about Raman spectroscopy, a Raman signal is what's called Stokes and anti-Stokes scattering. So what you typically see, or what most particles do, is Rayleigh scattering, which is they will absorb an amount of light. They will be excited to a certain state that corresponds to the energy of the light that they absorbed, and then they will fall back down to the original ground state and emit that light again. Rayleigh scattering is all over the place. It's the cause of the sky is blue and the sunsets are red and all that fancy stuff. So Raman scattering, Stokes and anti-Stokes, are both really weak signals. What happens there is they'll emit, or they'll absorb some light with a certain energy, go up to a virtual state, but then when they fall back down, they don't fall down to the original ground state. They fall down to another it's called a vibrational level, and they will emit light that corresponds to the energy that they fell and will still contain a certain amount of energy, which is this distance here that corresponds to the level that the vibration is. So these particles will still have a certain amount of energy in them, which is actually what we're measuring when we do Raman spectroscopy. So we're interested in what wavelength this energy corresponds to. And like I said, Raman spectra are typically very weak, so we need enhancement, which is why we do SERS and then photonic crystals on top of that to measure these spectra. So now that we've gone through the background, we'll go show some data. These are dark field images of different nanoparticles. So Claire can correct me, I don't remember which ones all these are, but these two are nanoflowers that she made. These are gold nanostars, and then these two are gold nanorods of different sizes. And dark field doesn't give you too much information about the size of the particles, but you can tell about the distribution. So for example, there's a lot of these gold nanorods, not so much of these. Yeah, the same with gold nanostars. There are a lot of them, but not a lot of the nanoflowers. And you can kind of see the arrangement that they lay themselves out in. So these nanorods are kind of clustered by themselves, and the nanostars all like to group together. So this is the reflection spectra from this data that I talked about. So this is just the reflection of the PC. And then these three are the resonance of the gold nanorods. So you can see the text is really small. These all have different resonance wavelengths, and it turns out that the nanostars, which is this one, the resonance was too high to couple with the PC. So if we go to the next, we'll go to this graph. These are the nanostars, which is why you can't see anything, because the resonance of the nanostars was too high to couple with the PC, so we don't get much enhancement. But the two different sizes of nanorods do work. This is using a Raman microscope, and then this is using the line scanner that I talked about earlier. So you can see they correlate pretty well, and we're actually getting good data. One other thing is these are the SEM images of the two different sizes of nanorods and then the nanostars. And Clara already talked about these earlier. See, the nanostars are huge, and the nanorods are, you can see that they're there, but they're tiny. And there were also, there were a high concentration of all three of them, so we're sure to get a signal. So a little bit of a summary. So we've talked about photonic crystals, SERS. Our work for the future is to continue to explore different shapes of nanoparticles and sizes and refine them into the ones that couple best with the photonic crystal. And we need to continue to work on the line scanner to find the optimum conditions for enhancement and to get good images. This is a picture of a photonic crystal. It's pretty colors. Yeah, so thanks to my advisor, Professor Cunningham. Thanks to Claire and her advisor for help on chemistry and giving me all the gold nanorod samples. And then thanks to my group and especially Renung for help on SERS and photonic crystals and getting the equipment up and running. Thanks to everybody in my group for thoughts and ideas, and then thanks to MCNTC for funding. So that's about it. If you guys have any questions, I'd be happy to take them now.